And thanks to the APS for organizing today's eChalk Talk. We're really excited to discuss, as Mary had mentioned, what we call mapping the missing link and really understanding and bridging this gap that exists between human genetics and disease biology. And we'll talk a little bit about how we go about doing this through small molecule biomarker discovery. As Mary mentioned, my name is Mohit Jain. I have the pleasure of speaking today on behalf of the Sapient team. And we're excited. So thank you to everyone for joining in on this beautiful summer afternoon. So one of the areas that we spend really quite some time thinking about is again, how do we go about bridging this gap that occurs between biology and drug development? And nowhere is this so obvious or such clearly a gap as it is with genome-wide association studies or GWAS work. And as many of you are aware, over the last 2 decades now, there have been over 5,000 GWAS studies that have been conducted. Over 3,000 traits have been genetically mapped, everything from height and BMI to age of death and mortality, lifespan, every virtual human disease, lots of circulating factors, and even such curious traits as IQ and baldness, if you will. And each of these has identified hundreds of common and rare genetic variants that are believed to contribute to the development of disease. Now, these have provided an enormous amount of information regarding sort of the genetic underpinnings, if you will, of various disease states, including synaptic pruning, for instance, in schizophrenia, autophagy in IBD, et cetera, et cetera. But if we really begin to examine this field now that's over 20 years old, the translation of these findings from initial GWAS work in humans to actual drug development have been more modest. And that really represents a fundamental gap that we have to be able to bridge in order to leverage this amazingly rich genetic information into drug development and improving human health. Now, the way we think about this, on one side, you have all of these tens of thousands of genetic variants that have been linked statistically, if you will, through these large human studies, everything from the UK Biobank to dozens of additional studies. And these are linked statistically to disease in a sort of a unequivocal manner in which we know these genetic variants, once again, are contributing to disease. But there's this missing component in the middle here that really allows us to take this information and use it for drug development. And this is biological insight. This is the basic mechanistic understanding that allows us to link genetic variants in human disease. And whereas parallel sequencing has massively transformed our ability to understand genetic variants and begin to associate them with human disease biology, biological insight has really trailed in its acceleration. And this serves as sort of the fundamental missing piece that allows us to connect human genes on the left side with disease processes on the right. Now, there's no unifying simple solution for beginning to link these two, but we would propose today that small molecule biomarkers can be absolutely instrumental in allowing us to form this link between genetic variants and disease biology. And what we're hoping to do for the next 15 to 20 minutes or so is really talk through our work in small molecule biomarkers, what exactly these represent, and how they may allow us to begin to understand insight into underlying genetic variation and ultimately translate this information to better drug development. Now, just to begin with, what are actual small molecule biomarkers? Now, these are the tens of thousands of little factors that are circulating in human blood. And what make these very special in relation to genetic factors or protein biomarkers or other types of biomarkers that we typically think of is that these small molecule biomarkers read out both genetic as well as non-genetic factors that contribute to human disease. Now, we can think about both normal tissue, disease tissue, as well as even the microenvironment, if you will, in the case of cancer or whatever the disease may be. And in each case, these small molecule biomarkers are being produced intracellularly in response to a number of signaling pathways or biological mechanisms. Now, given their physical chemical properties, which are quite a bit different than DNA or proteins, these small molecules are actually actively extruded from the intracellular space into the blood. Now, what this allows us to do is to be able to massively assay these molecules in the circulation, and in doing so, understand what exactly is occurring at the level of the tissue. Again, whether it be for the normal tissue, for diseased tissue, or even microenvironment tissue. And this is a very special property of these small molecule biomarkers, and we'll show you some examples of how we go about doing this in just a moment. The second aspect of these small molecule biomarkers that's really quite special is, given the fact that these are among the most ancient of molecules that are present in humans, is that they're highly conserved, both from in vivo states in humans all the way down to preclinical models and even in cellular systems. And this provides a very sort of unique opportunity to measure these molecules across these multidimensional modalities. Again, whether it be cells in a dish as part of a screening assay, whether it be in preclinical models, everything from mice to rats, non-human primates, dogs, etc., and then ultimately in large-scale human populations. Now, the challenge to date has really been around how do we actually go about measuring these small molecule biomarkers? And particularly, how do we do this at scale in large human populations? And this is fundamentally what's required in order to be able to use these types of information to bridge human genetics and disease biology for the purpose of drug development. And this is where Sapient comes in. Sapient has developed what we consider next generation ultra high throughput rapid liquid chromatography mass spectrometry, or RLCMS technologies. Here's an actual picture of our suite of high throughput mass spectrometers at our Sapient site here in San Diego. And essentially what these new technologies allow us to do for the first time is measure thousands of these molecules and thousands of human biosamples at a given time. Now, to give you a little bit of more granularity on what exactly we're capturing, these instruments have the sensitivity and capacity for what we call non-targeted mass spectrometry measure of over 11,000 small molecule biomarkers. These assays can be performed in a relatively modality sort of agnostic manner, meaning in plasma and serum and urine, in tissue samples and tumors, again, both from preclinical as well as clinical systems, all the way down to cells in a dish, media, and even lower organisms such as bacteria and yeast. For each of these molecules, or these 11,000 or so small molecule biomarkers that are captured, we're able to do what's called 4D chemical classification. And what this refers to is the fact that we're able to capture a number of physical chemical properties for each of these molecules, including their retention time, their MS1, what's called their MS2 patterning, meaning when these molecules are fragmented, we can capture all the mass information regarding their daughter fragments, and then what's termed the CCS value, or their physical chemical properties on ion mobility mass spectrometry. Now, this is all done in a relatively simultaneous manner across broad chemistries, everything from very apolar molecules to very polar molecules, from organic acids and amino acids, all the way through lipid-based molecules, both bioactive polar lipids as well as neutral structural lipids. Now, we can do this really in quite rapid a fashion. Our analytical cycle time is just under 53 seconds per sample. And what this really allows us to do then is have the capacity to handle tens of thousands of biosamples at a given time, and perform these analyses at greater than several thousand samples analyzed per 24-hour period. And ultimately, this is the throughput that's required in order to be able to perform true population scale analysis. Now, what makes Sapient quite special is not only have we developed these technologies, but already we've performed these analyses on hundreds of thousands of biosamples from around the world. And as I'll talk about in just a quick moment, these have all been integrated in what we call our human biology database that serves as an incredibly rich repository of circulating small molecule biomarkers in relation to human phenotypes and genotypes. And we can leverage this information for beginning to link, as I mentioned initially, genetic variants to human biology. Now, these analytical technologies just represent one of the three core components of Sapient and our approaches. The analytical technologies, again, are shown here at the top. These are these next generation proprietary mass spectrometry systems that allow for measure of thousands of these molecules and thousands of biosamples at a given time. Now, these technologies have been integrated into two other core components. The first is a biocomputational, integration system. Now, what this comprises of is an expert data science team that has deep expertise not only in the handling of mass spectrometry data, but also much more advanced modeling and machine learning that allows us to integrate mass spectrometry data together with clinical measures, other circulating factors, genetic factors, as we'll show you in just a moment, protein and proteomic factors, as well as many, many other multidimensional data in order to derive true actionable insight regarding core molecules that influence human disease biology. And again, we'll show you an example of this in just a moment. And the third component of Sapient's approaches, as I mentioned, is what we term our human biology database. And this is a very, very rich data repository that now consists of hundreds of thousands of human biological samples that have been collected from around the world and analyzed using our technologies and our biocomputational integration. And this is a very, very rich database, both of genotypic information, protein markers, cytokine measures, as well as clinical outcomes that allow us to begin to use the information that's derived here for true discovery. Now, as I mentioned, the real goal here is to use small molecule biomarkers to bridge the gap between large-scale genetics data that largely exists now in the public and scientific domains and biological understanding. And it's this key biological insight or biological understanding that's required in order for us to be able to take genomics and genetics information and leverage this for drug development. Now, there's three fundamental ways in which small molecule biomarkers can contribute to this underlying process. The first, is shown here on the left, is that small molecule biomarkers can inform on biological mechanisms linking genetic variants in human disease, essentially providing insights into the actual mechanisms by which these variants may influence disease processes. And we'll talk about examples of that in just a moment. The second is, not only do these small molecule biomarkers provide information regarding natural genetic variation, these can also be fundamental in providing information regarding readout of genetic targeting, whether that be a small molecule genetic targeting all the way through CRISPR and RNAi-based approaches for genetic targeting, and essentially read out target engagement for these types of manipulation studies. The third way in which these small molecule biomarkers can be instrumental is for allowing for discovery of disease-relevant pathways. And again, we'll talk about what this means, but essentially beginning to take all these thousands of genetic variants and organize them into pathways or common modalities that contribute to human disease processes, which is really essential for understanding and beginning to make sense of the massive amounts of genetic data that's available out there in the world today. Now, how do we go about do this? And particularly, how do we map sort of the relationship between genetic variants and circulating biomarkers, and in doing so, provide biological insight? Now, the way we go about this at Sapient is through leveraging of an approach that we call metabolic quantitative trait loci, or MQTL analysis. And essentially what this consists of is, in our human biological database, we have tens of thousands of individuals in which we have captured not only blood samples for analysis using our mass spectrometry systems and capturing of tens of thousands of what we call these small molecule biomarkers on the left here, as well as in these same individuals, we have underlying genotype. And particularly imputation around 20 million common disease SNPs in these individuals. And by having these same data sets in the same individuals, both small molecule biomarkers as well as genotypic information, we can begin to put these information sources together, and in doing so, essentially perform genome-wide association studies for each of thousands of different circulating small molecule biomarkers across these tens of thousands of individuals. And in doing so, we can begin to understand how do normal common genetic variants, whether they be normal variants or disease-causing variants, actually associate with circulating biomarkers. Now, this is what it actually looks like across the human genome. So this is a modified Manhattan plot. If you look at the x-axis, it provides a chromosomal loci information. On the y-axis here is the negative log 10 p-value. And it essentially begins to associate the thousands of circulating small molecule biomarkers that are captured by our systems with common genetic single nucleotide polymorphisms across tens of thousands of people. Now, there's a couple things, and we'll talk about the granularity around this data in just a moment, but just on a global scale, there's a couple interesting things you see here that just jump out on initial view. The first is that they're genetic hotspots, if you will, that represent these spikes, which by their circulating loci, there's particular genetic loci that are highly associated at p-values north of 10 to the minus 100 with particular molecules. Now, some of these genetic loci are associated with a single molecule, as you see for these blue spikes, whereas there are other genetic loci that are associated with tens to hundreds of different circulating molecules. Now, to give you an idea of what this may represent, you can imagine that common genetic variants in the circulating, in the cytochrome P450 system, for instance, those genetic variants may influence the production and/or biosynthesis and/or detoxification, if you will, of tens of thousands of different molecules in human blood. And those are genetic variants that may influence many, many different circulating small molecule biomarkers. In contrast, there are other genetic variants which only associate with one or two biomarkers. And both of these can provide very interesting and important information that, again, allow us to begin to link human genetic variation and disease. Now, if we look at the summary information for these analyses, this is what we find. Just under half of all the molecules that we measure in people, somewhere in the order of 60 to 70,000 small molecule biomarkers, almost half of these are strongly associated with at least a single genetic loci. And again, these are what we consider genome-wide statistical thresholds of 10 to the minus 6, 10 to the minus 7. Now, if we look at all the SNPs, all the common SNPs that are present in humans, and we have imputation of among 20 million or so common genetic variants in the human genome, we find that just north of 5% of total SNPs are associated with at least one circulating biomarker. Now, if we look at those SNPs that actually occur within the transcribed gene region, so the exonic region, and particularly focus this analysis on a gene level rather than on a SNP level. So if we look across the 19,000 or so genes that are part of the human genome, we find that for greater than 90% of these genes, again, this is an analysis performed on a gene level as opposed to a particular SNP level, we find that there's SNPs within the transcribed gene region that strongly associate with at least one circulating small molecule biomarker. Now, this is incredibly powerful. It means for more than 90% of genes in the human genome, we're able to read out natural genetic variation through the circulating small molecule biomarkers. And you can imagine how powerful this is for beginning to translate human genetics into disease and drug development biology. Now, not only, as I mentioned, can we read out natural variation that occurs in these underlying genetic variants, but also you have the power here to now read out genetic targeting, whether these be done through pharmacologic targeting of a particular biological pathway, all the way through CRISPR-based genome editing of specific genes. And so in the same way, we can read out normal variation, we can read out sort of disruption, if you will, of a particular gene, both through direct target engagement biomarkers, as shown here in red, as well as through secondary indirect or pharmacodynamic markers that may be shown here in blue. And once again, given the physical chemical properties of these small molecules, they can be produced intracellularly and following genetic targeting, again, through pharmacologic and/or through genetic means, these small molecules can be released from the intracellular space in the given tissue into the blood, where this information can be captured as a readout of biological processes, whether it be activation and/or repression of specific genes or biochemical pathways. Now, the other way in which these large-scale analyses can be really sort of insightful is in organizing human genetics data. And anyone who's worked in this space know that for most common diseases, again, whether it be heart disease, diabetes, obesity, cancer, neurodegenerative diseases, etc., there are hundreds to thousands of different genetic variants, each of which has a relatively small effect on underlying phenotype. Now, organizing this information into biological pathways has really been challenging, and this is one of the big limitations that's prevented drug development in this space. And this is where having these circulating small molecule biomarkers, again, that map to more than 90% of the genes within the human genome, can be absolutely critical. And one can begin to use this information to begin to organize these genetic variants into 3 to 4 to 5 different biochemical pathways or biological pathways, all of which are influencing a particular disease phenotype through a shared mechanism. And this is a way in which we can begin to provide some insights and understanding into particular groupings of genetic variants. Now, let's just take in the last couple minutes here, just a moment to talk through some real world applications and how we can actually do this in large data sets. And I'm not going to belabor this. I want to make sure we leave enough time for questions. But one sort of biological pathways you can imagine that we've been quite interested in is the PD1, PD1L pathway. I know this is a pathway that did not emerge from human genetics, but rather from fundamental biology. And has really been the mainstay of cancer therapeutics over the last several years here, and particularly targeting the immune system to reawaken in order to target cancer directly through modification of this PD1, PD1L pathway. Now, if we look at common genetic variants in both PD1 and PD1L, and again, look across the tens of thousands of small molecule biomarkers that are captured in our systems and our human biological database. Again, in this modified Manhattan plot, that's what's shown on the x-axis. It's the number of biomarkers. What you see here is that there's 5 to 6 different circulating biomarkers here that strongly associate with underlying genetic variants in the PD1 pathway. Now, these have been verified in multiple independent data sets in our hands across broad ethnicities and underlying genotypes, and that's one of the underlying powers of the diversity of our underlying human biological database. Now, if we look at any one of these circulating markers and ask, well, how does this actually read out manipulation of this pathway, particularly pharmacologic manipulation through large molecules, what we find is super interesting. So if we look at individuals before and after they receive PD1L treatment, what you find is that, again, just a single biomarker here that was discovered in the prior GWAS analysis, what we can find is that these markers change from pre to post treatment. And so they can read out not only genetic variation, but also dynamic interactions, as I mentioned, and target engagement of the PD1L system. And most importantly now, if we actually take this information and look in large clinical studies that have employed PD1 across a number of different solid tumors, what we find is really fascinating in that those individuals that increase these biomarkers after receiving a PD1 therapy are much more likely to respond to an actual PD1 drug relative to those individuals that have low levels of these pharmacodynamic markers after receiving therapies. And so this is just one such example by which we can begin to discover circulating small molecules that are reading out this fundamental biological pathway using human genetics, and then leverage this information as part of the drug development pipeline. Again, both here discovering a pharmacodynamic marker that's changing in relation to receiving the PD1 therapy, and then ultimately use this to enrich those individuals who are going to be responders versus non-responders. Now, this is just one such example. As you can imagine, there's hundreds such examples here that we've sort of internally mined within Sapient, whereby we're beginning to use these tens of thousands of circulating small molecule biomarkers to begin to understand the link between human genetic variation and human disease for the sake of drug development. I'll go ahead and stop there. I want to make sure we leave enough time for some questions. For my last slide here, this is our contact information to schedule a time to chat with us. Our email is discover@sapient.bio. We also have a Calendly link here and a phone number, as well as our social media sort of sites, if you will, on the bottom. And we'd be really excited to chat with anyone who may be interested in learning more about these approaches and thinking through how we may be able to use these circulating small molecule biomarkers to accelerate their drug development pipelines. I'll go ahead and stop there, Mary, and thank you everyone for listening for the last 20 minutes or so, and I'd be excited to take any questions.
Thank you, Dr. Jain. We do have a few questions, and I'd like to pose them to you. How do you begin to link biomarkers associated with genetic variants causally to disease?
Yeah, it's a great question, Mary. And so one component here is really thinking through the genetic links, which is linking genetic variation with disease biology. And if we're putting small molecule biomarkers in the middle of that as a means for understanding biological insight, how do you begin to link the biomarkers themselves to disease, particularly in a causal manner? Now, there's a couple different ways we go about doing this. As mentioned, for this human biological database, we have now hundreds of thousands of individuals, including with longitudinal outcomes over 10 to 30 years in these individuals. And by profiling these molecules and these biomarkers in their human blood, as well as having adjudicated clinical outcomes in these individuals, we can begin to do association between particular biomarkers in blood and the development of disease. Now, by also integrating underlying genetic variation and other sort of data sets within this process, one can begin to sort of infer causality using Mendelian randomization, as well as a number of other processes. And we do this quite routinely as a way of beginning to understand circulating biomarkers and their causal link to disease processes.
All right, thank you. Another one asked, can you further discuss how these circulating biomarkers can become part of a drug development program?
Yeah, it's a very good question, and this is one of those that there's no one size fits all. As you can imagine, there's many ways in which we can begin to leverage these small molecule biomarkers to accelerate and advance forward drug development. And I can give you just some quick examples by which we can begin to do this and how we think about this. So for instance, as we've talked about today, these small molecule biomarkers can provide a tremendous amount of biological insight into how human genetic variation may be influencing disease processes, allows us to organize this information, may provide insights into underlying target engagement. But there's many other ways in which these small molecule biomarkers can be absolutely critical as part of a drug development pipeline. These can be instituted anywhere from very early steps in the discovery phase when targets are being elucidated, all the way through the preclinical sort of steps, and including clinical studies all the way from phase 1 to phase 4. And so it's through the entire drug development pipeline that we leverage these approaches, and they allow us fundamentally to begin to link the right patient with the right disease process and a specific therapeutic. And you can imagine that occurs throughout that pipeline. And so, for instance, there are clients we work with to make these small molecule biomarker measurements to understand new targets, as well as to understand target engagement for particular pharmacologic therapies, as we talked about today. There's other clients in whom we work with to understand companion diagnostics and identify biomarkers of responders versus non-responders, or to even rescue failed therapeutics, or to understand addressable markets or early diagnostic biomarkers for particular disease processes. And so we really fundamentally think of these as tools that can be applied in a host of different manners, depending upon the underlying question throughout the spectrum of drug development, if you will.
Okay, one more. Can you speak more on how small molecule biomarkers can be used to identify druggable targets?
Yeah, absolutely. Well, as you can imagine, oftentimes in the case of human genetics, it's not always sort of the genetic variant or the particular gene that's a variant, I should say, that represents the ideal drug target. And these genetic variants can influence a number of biological pathways that can be read out by these small molecule biomarkers in human blood. And in this way, we can oftentimes begin to discern what is the actual ideal target itself. Not only what is the correct sort of protein to modify as part of disease mitigation, but also to take these small molecule biomarkers now and understand target engagement around that specific marker, or pharmacodynamic measures, if you will. And so this is a way in which these small molecule biomarkers can really help identify what are those pathways that are being modified within a common disease process, and how we can therapeutically begin to sort of attack these, if you will.